In this video, I will examine the effect of electrical stimulation on the force of contraction of skeletal muscle, as demonstrated by Dr. D on a simulation on YouTube. The force of a muscle contraction depends on four factors: the number of muscle fibers recruited, the size of the muscle fibers, the frequency of stimulation, and the degree of muscle stretch. This study examines the effect of the number of muscle fibers recruited and the frequency of stimulation on force of contraction in a skeletal muscle. Let's first examine the anatomy of the skeletal muscle. Muscle fibers are found inside of muscle fascicles, which are found in the skeletal muscles. Inside the muscle fiber, multiple myofibrils exist. These contain the contractile units of the muscle. The functional contractile units in each myofibril are cylindrical bundles called sarcomeres. The sarcomere contains thin filaments and thick filaments, which slide over each other during muscle contraction. The myosin forms the dark A bands, while the actin forms the lighter I bands, giving the muscle its striated appearance. Each sarcomere contains a Z disc at each end, where the actin myofilaments attach. The sarcoplasmic reticulum forms a mesh around each myofibril, and extensions of the sarcolemma wrap around each myofibril, forming T tubules, which are flanked on each side by sarcoplasmic reticulum forming triads. Finally, each muscle fiber is innervated by a motor neuron, forming a motor unit. This neuromuscular junction is found on the surface of the sarcolemma in an indented region called the end plate. This is where the muscle fiber first receives the stimulus to contract from the motor neuron. Skeletal muscle contraction starts when an action potential from a motor neuron causes depolarization in the membrane of the sarcolemma of the muscle fiber. The depolarization spreads through the muscle fiber through the T tubules. The depolarization causes the opening of receptors on the sarcoplasmic reticulum, where calcium is stored. When the calcium is released, it binds to troponin. This causes a shift in tropomyosin, which allows the myosin heads to attach to the actin filaments, creating a cross bridge. When ATP, the energy molecule, binds to the myosin head, the myosin dissociates from the actin, which breaks the cross bridge. The ATP is metabolized into ADP and phosphate, causing the myosin heads to change shape and move down the actin. When the ADP is released, the myosin goes back to its original position, pulls on the actin, causing the sarcomere and the whole muscle fiber to contract. Muscle contraction stops when the signal coming from the motor neuron stops, which repolarizes the sarcolemma and T tubules. This causes the voltage-gated calcium channels in the sarcoplasmic reticulum to close, which causes calcium ions to be pumped back into the sarcoplasmic reticulum, causing tropomyosin to recover the binding sites on the actin. Muscles can also stop contracting when the available ATP has been exhausted, which causes them to become fatigued. The purpose of this study was to demonstrate the effects of electrical stimulation on skeletal muscle contraction. Single stimulation was run at multiple voltages to identify a maximum stimulus. Then, multiple voltages were applied in a stimulus-rest stimulus pattern, a back-to-back stimulus pattern, and a four stimuli pattern to identify the effects on the force of muscle contraction. Finally, the muscle was stimulated at 50 stimulations per second and 130 stimulations per second to evaluate the effects on muscle contraction. These computer simulated experiments were performed by Dr. D on the ANP1 Muscle Physiology Part 2 YouTube channel, and all data comes directly from there. In the simulations, a muscle cell was suspended from a vertical apparatus as shown in the image. The muscle was held in place at the top and the bottom, and electrodes were attached to the muscle. The electrodes were attached to a virtual power source and a virtual voltmeter, which was linked to a computer. The simulation was run in three parts. In part 1, voltages that simulated action potentials were applied to the muscle at 0 volts, 1 volt, 2 volts, all the way through 9 volts. As each voltage was applied, the force of the active muscle contraction was recorded in grams, and a visual representation of voltage versus force was provided. In part 2, an 8.5-volt stimulus was applied to the muscle, and multiple muscle contraction patterns were manually simulated on the muscle. A stimulus rest stimulus pattern, two back-to-back stimuli without returning to baseline in between, and four back-to-back stimuli without returning to baseline in between. The resulting forces of each contraction pattern were recorded along with a visual representation of voltage versus force. In part 3, multiple stimuli were delivered at 8.5 volts at a rate of 50 stimuli per second and at 130 stimuli per second. The resulting forces of each contraction pattern were recorded along with a visual representation of voltage versus force for each. Part 1 of the experiment demonstrated the effects of increasing voltage applied to the muscle with the voltage simulating action potential. As seen in the graph of the data from the simulation, we see that the force of contraction increased as applied voltage increased from a low of 0 grams at 0 volts to a maximum of 1.82 grams at 8.5 volts. This is visualized by the peaks on the graph. When the applied voltage was increased to 9 volts, the force of contraction remained at 1.82 grams, demonstrating a maximum stimulus of 8.5 volts. The force of contraction does not increase with increasing stimulation levels, indicating that all available muscle fibers are contracting at a stimulus of 8.5 volts. Part 2 of the experiment demonstrated the effects of three stimulation patterns on force when stimulation was applied at the maximum voltage of 8.5 volts, as demonstrated in Part 1. This figure shows the results of a stimulus rest stimulus pattern. Here, the muscle was stimulated at 8.5 volts, allowed to return to baseline, and stimulated again. The results show a force of 2.41 grams and also showed that applying a second force after returning to baseline yielded a higher force in the second contraction than in the first, higher by 0.59 grams. This graph shows the effect of the application of two back-to-back stimuli that were applied without returning to baseline in between. This gave a force of 2.79 grams. This was 0.97 grams higher than the single stimulus at the same voltage and 0.38 gram higher than the stimulus rest stimulus condition. In this condition, four back-to-back stimuli were applied to the muscle without rest at 8.5 volts. This yielded a force of 3.66 grams, which was 1.84 grams higher than the single stimulus at the same voltage and 0.87 grams higher than the two back-to-back stimuli condition. In the second demonstration, the effects of multiple stimuli on the muscle are seen. In all three conditions, stimulus, rest, stimulus, two back-to-back stimuli, and four back-to-back stimuli, the active force generated when a maximum voltage of 8.5 volts was applied was larger than that of the single contraction alone, 1.82 grams, at 2.41 grams, 2.79 grams, and 3.66 grams, respectively. These results indicate that when a skeletal muscle is repeatedly stimulated, muscle twitches will overlap each other, giving an additive effect in force of contraction in a phenomenon known as wave summation. This is clearly seen in the 0.59 gram increase in force between the single stimulus in part 1 and the stimulus rest stimulus condition in part 2, showing that even when a muscle is allowed to relax briefly between contractions, the second contraction will be stronger. The 0.97 gram increase in force between the single stimulus at 8.5 volts and the two stimuli when the muscle did not return to baseline between contractions shows a larger wave summation effect. Finally, the 1.84 gram increase in force between the single stimulus at 8.5 volts and the four stimuli when the muscle did not return to baseline between contractions shows an even larger wave summation effect. Wave summation was achieved by increasing the frequency of the stimulus to the muscle. In part 3 of the experiment demonstrated the effects of rapid continuous stimulation applied at the maximum voltage of 8.5 volts. The figure on the left shows the effects of 50 stimuli per second being applied to the muscle. The measured force was 5.2 grams. On the right, we see what happened when 130 stimuli per second were applied. The force was measured at 5.88 grams, a force that was 0.68 grams higher than the 50 stimuli per second condition and 4.06 grams higher than the single stimulus applied at maximum voltage. The loss of wave activity seen when these multiple rapid contractions of 50 and 130 stimuli per second were applied to the muscle suggests that the muscle is in a state of near tetany, where the muscle remains in a contracted state. The loss of wave activity is more pronounced as the stimuli become more frequent.